Hello, everyone. Thanks for coming today. As Gabe said, I'm going to be presenting my research on intracerebral transplantation of autologous bone marrow stem cells, which produce functional recovery in rats with long-term strokes. So stroke is the fifth leading cause of death in the United States. About every 40 seconds, someone in the United States has a stroke, and about every 3.5 minutes, somebody dies from a stroke. Every year, around 800,000 people in the US have a stroke, and about 600,000 of these are new strokes, with 200,000 of those being recurrent. Nearly 1 in 4 people have had a previous stroke for those that have had a stroke. About 80% of those strokes are ischemic, and this costs the United States around $53 billion per year, and that number is only rising according to the CDC data. So there needs to be new and improved therapies for this in order to treat this growing population. So current treatments are limited. Throughout the past 2 decades, the management of stroke has changed, shifting from an approach limited to secondary prevention to one focused on early prevention strategies. So we really only have 2 options. Our first option is treating with an IV medication called tissue plasminogen activator, which can be injected into the patient and can go into the brain and break up the clot that's causing the stroke. This can only be given within about 4.5 hours of the onset of a stroke. So it's very limited, and it can only be used for a handful of patients that are able to get to the hospital quickly. If those patients aren't able to get to the hospital immediately, intra-arterial mechanical thrombectomy is able to be used. This is the picture on the right showing what that is. Basically, a catheter is threaded through the leg up to the brain, where surgeons can remove the clot using these coils, which can essentially grab onto a clot and pull it out. The issue with this is that it can only really be done within 24 hours of onset of a stroke. A large number of patients that have strokes actually don't even know that they're having them, especially the ones that live alone or elderly, might not have people around them. So identifying a stroke is important, but for those people that are not able to get to the hospital within a day of having this stroke, new therapies are needed. So what about the rest of the patients? There are not treatment options for around 85 for a lot of these patients, and that's around 85% of the patients that come to the hospital with stroke after 24 hours. Over the past 2 decades, this has led to stem cells as a potential therapy. Although initially designed as a replacement therapy, mesenchymal stem cells are not thought to replace missing neural circuits. They seem to act more as modifiers of the environment, changing the inflammatory landscape of the brain, increasing levels of anti-inflammatory cytokines, growth factors, and decreasing pro-inflammatory cytokines. So over the past 2 decades, there have been numerous studies on mesenchymal stem cells as a treatment for stroke in a rat model, the MCO model, which I'll get to in a moment. Most of these published studies looked at a handful of different factors, whether it be allogeneic stem cells, meaning other, so from a different animal, cells that were harvested from a different animal, or different routes of administration. So that could be intravenous or intra-arterial. And so not all that much research has looked at intracerebral injection directly into the brain, as it's a little bit more invasive, but perhaps that might be more effective. So there are 2 clinical trials currently that have been completed. One in out of Stanford and one out of I think it is Texas. Both of these were genetically engineered cells. One was given Carotid injection, so through the neck, and the other one was given directly into the brain. These are very recent, within the last couple of years. The one on the left is the one that was given intra-arterially, and they did not see as much recovery as they would have liked, while the ones given directly into the brain of patients, it was a lot more effective, although these were genetically modified allogeneic, meaning of other people, of a donor. And so, more studies are needed to see perhaps we could get around a lot of these limitations if we could deliver the stem cells directly from the patient itself, their own stem cells. So one of the issues with the previous studies looking at intra-arterial intravenous injection is that these cells are large and they don't get to the brain. So a low amount of them actually make it to the brain. Most go to the lungs, the liver, the kidney, or the spleen. So, for example, here's one previous study that was done in 2011, where intravenous injection was very inefficient. As you can see here on the right, most of the cells actually made it to the spleen, lungs, liver, and kidney. Some made it to the brain, but they all sort of filter out very, very quickly. And showing from the graphs on the left, which were some behavioral analysis, you can see that there really wasn't a significant recovery when given, and that's definitely due to the quick clearance from the brain, as well as the small number that actually made it through the blood-brain barrier. On the other hand, some intracarotid injection has been done. There was some functional recovery, but there was still rapid clearance from the brain, showing that perhaps there wouldn't be as much sustained recovery if it were to be continued out. And so this was a paper published back in 2010, but like I showed you on a previous slide, when this was repeated in humans, it didn't really work. So that's really forced us to look at other avenues of administration. So once again, just to go over the blood-brain barrier, very limited capacity to allow mesenchymal stem cells through it. Capillaries are very small, around the diameter of a red blood cell. A mesenchymal stem cell is much larger than this. And not just that, but these capillaries are much smaller than the mesenchymal stem cell itself. So really, injection through the veins or through the arteries will most likely prove, what we've seen is not proven too effective, and will most likely continue to not be as effective going forward. And so that led to our current study. So our study here in Philadelphia at Thomas Jefferson University, we are looking to intracerebrally transplant autologous mesenchymal stem cells into stroke patients. We started by doing this in the lab in a rat model. And so we wanted to first look at what was our best dose. Other studies had published dosing on the lower side, 1, 2.5, and 5 million cells should be somewhat sufficient, especially if we don't have to go around the blood-brain barrier. Looking to see where these cells might end up. We also want to see how long do the cells actually stay there in the brain, going along with where do they go? And then does this actually change the size of the infarct of the stroke, the dead tissue in the brain? Do these cells regenerate? We also wanted to know, is there functional recovery and behavior change? Do these animals actually get better following administration of mesenchymal stem cells? And lastly, do these stem cells alter the glial reactivity and inflammation within the brain? So this is the outline of our study. On day 0, or day here -29, we start with our MCAO, which is the middle cerebral artery occlusion. What we do is we insert a filament into the rat's carotid artery up to the middle cerebral artery. We leave it there for 2 hours, and that allows blood flow to that area in the brain to be blocked. And then after 2 hours, we remove that filament, which administers a stroke to this animal. The following day, we were able to do some behavioral testing, as well as an MRI, to assess the level, the size, as well as the severity of the stroke. A couple of weeks after that, we are able to take bone marrow from the rat's tibia and plate it on a flask. And then a week after that, we are able to grow it in our sterile growth bioreactor in order to grow large quantities of these cells rapidly and cleanly and sterilely. About a week after we grow these cells in the bioreactor, we are able to do another MRI and some behavioral scores to get a day 0 assessment. We're able to do flow cytometry on the cells in order to assess, to make sure that these cells are not differentiating to their translineage, into their downstream translineage markers. And once we have our MRI, we can then see where we want to place the cells. Our goal is to place them around the infarct, around the dead tissue. And then we can set the animal up for transplantation. And then 1 week, 4 weeks, and 8 weeks following transplantation of these cells, we can continue to do MRI and behavior in order to assess the level of recovery and the change that might be in size of infarct. And then 8 weeks following our transplantation, we can sacrifice the animal and take a look at the histology of the brain and do some postmortem analysis. And so I also wanted to just show this is our bioreactor. Basically, what this allows us to do is we can take small quantities of bone marrow from a human, or in the case of a rat, we had to grow it up on a flask early in order to get enough cells, just because there was such a small volume from the animal. And this is a closed system. There is no contamination whatsoever. We can hang a bag of media, we can hang bags of wash buffer. This connects to gas, and it allows us to regulate cell growth. And so on this, if I can get the pointer up here, you can see this small part in the middle. This is actually the bioreactor core itself. And so we're able to inject the bone marrow directly into there, put in a protocol telling the machine how much, how fast the media needs to go through for how long, with how much gas. And then after, about a week, we can tell the machine to harvest the cells, release the cells from the core of the bioreactor, and we can get a full bag of mesenchymal stem cells. And so before I get into talking about the results of our study, I think there is an audience poll that I think Gabe will take over for a second.
Thanks, Max. Thanks for the great discussion so far. And as Max mentioned, before we continue, I would like to ask a couple of quick poll questions. The questions would be the following. So the questions are, what is the main focus of your research? And would you consider implementing MRI into your research? Please select the most appropriate response, and we will wait a few moments to let everyone answer the questions. We'll just wait a few moments more. Thank you for answering the poll questions. I will now hand over the presentation back to Max.
All right, thanks, Gabe. All right, so continuing on. The first bit of results that we compiled and want to present here is functional recovery of the animals following transplantation. So like I had mentioned, we transplanted the animals at day 0. So this was a month after the stroke itself. And that's an important part that I glossed over, is a lot of these previous studies looked at giving these cells quickly after the stroke, within a day, within a week. We're looking at a month, a stable stroke. These are for patients that have gone a long time after their stroke. They did not receive the tissue plasminogen activator, the TPA. They did not receive a thrombectomy. They don't really have any other options. So we're assessing a long-term stable stroke. And so what we found here was Within a week, essentially, at all three groups where we looked at 1 million cells, 2.5 million cells, 5 million cells administrated, there was a highly significant change in behavior. So this is looking at the Modified Neurological Severity Score, the MNSS, which assesses behavior based on a handful of parameters. I've got two pictures here showing on the right. You can see the rat doing the grasping test. So this assesses, can they grasp a bar and can they grasp it tightly? And then on the bottom here, you can see the lateral movement test, where you kind of push the animal back and forth and see if he can push back against your hand. Is there a strong force against you? And so what we found here is really compelling, that these animals that at day 0 started with a score around 11 or 10 really seem to get better down to around a 5 or 4 or 5 by day 60 following administration. It's interesting too, because when we look at this graph, you can see that there isn't a plateau. It seems like if we continue this on maybe to 80 days, maybe to 100 or 120 days, would they have recovered to a 0? So that's an interesting bit that we didn't look at, but it's something to think about going forward. So next, we also wanted to assess lesion volume using our brand new MRI. And so what we saw here was that there really wasn't much change to the size of the infarct. I do want to point out that in this group 2, it looks like there was a huge change. We're not super convinced in that. We think that perhaps it was because it was a small number of animals. There were 6 animals per group. We wonder if maybe we bump the number, the N up, we would see, we would see something similar to group 1 and 3, but perhaps this is the correct dosage. We don't assume that this is really changing the landscape of the brain that much. We've got some images here on the left showing 2 different MRI sequences, the T1 weighted image, which is the darker of the 2, and the T2 weighted image, which is the lighter. You can see on the top 2 rows, we have our control animals. On the bottom 2 rows, we have our one of our groups. You can see there's really not much change in either group. And it looked the same for that same group 2 and group 3. Looking at day 0 in this first column, 7, 30, 60, as we move along, you don't see much change. And if you look at the T1s, that darker shaded region on the right side of the brain or the left side of the image, or on the T2, the very bright hyper intensity, you can see that's the dead tissue. You can very clearly tell that a large portion of that brain has essentially disintegrated following that stroke. And so basically, we didn't see a whole lot of change, and that's similar to what other groups have shown. So another interesting bit about this is we wanted to look at exactly where are the cells going, after we inject them. Are they sticking around, and are they, moving around within the brain? And so we did another study using a whole different set of animals. We basically replicated the study. We used 3 animals per group. And the difference in this study was we did not use the bioreactor, and we aspirated the cells, the bone marrow from the animal. We grew them up on a flask. And 1 day prior to injection, we gave these cells what's called quantum dot nanoparticles. They're these tiny little nanometer sized particles. They have a fluorescent tag on them. The cells are able to endocytose them or take them up, and they are able to fluoresce when we look at them under a microscope. So looking at the first thing at the graphs, we can see that we see essentially a similar, even at the small end, we see essentially a similar behavioral affect in these animals, which is very it's important because it concurs to what we saw before in the overall study. Looking at the bottom 3 images, we can actually see these are 3 different animals that had gotten the stem cells. You can see it's 7 days, 30 days, and 60 days. You can see the hole where the stroke had occurred. And if we zoom in a little bit more, we can actually see the stem cells still there. And what's really interesting is that we can see the stem cells there at 7 days, 30 days, and 60 days. So those cells are not leaving, they're sticking around. Previous work using allogeneic stem cells showed that these cells cleared out from the brain pretty quickly within a week. And the same goes for the ones that were injected intraarterially or intravenously. But what we're seeing here is that these cells are sticking around for quite a while. And actually here on the right, we have an even higher powered image where the blue is DAPI, that is our nuclear marker of the nucleus. And then around it, we can actually see our nanoparticles surrounding it. So we have confirmation that these cells are taking up this dye, these nanoparticles. And that's really giving us a good ability to visualize these cells once we are able to look at the histology of the brain. So once we realized that these cells were sticking around for so long, we got a little bit nervous because other people have not reported this. And when you have stem cells in an area that are staying for so long, not changing, we do get nervous that, our first thought is, cancer. We don't want to put in stem cells that could then become oncogenic, to mutate to oncogenic cells. So the next thing we looked at was, do these cells turn into cancer? Can they divide? Can they mutate? And so what we did was we stained these cells for a marker called Ki67, which is a cell cycle marker. It's largely used in cancer research to detect, growing and dividing tumor cells. You can see on the top row, this is one of our animals that have received the quantum dots. You can see around the cells, and there was no staining for Ki67 anywhere. And this goes for all 9 of the animals that were included in this QDOT study. And then on the bottom, we can, using a positive control as MSCs in culture that were rapidly dividing, you can see there was a large amount of this Ki67 in it. So this confirms to us that, even at 60 days, these cells are not dividing and they're kind of just sticking around. So another interesting point we wanted to look at is, are they changing? So not only are they growing and dividing, but are they changing? Are they becoming neurons or astrocytes? So we stained with S100 beta and beta 3 tubulin. We were able to look at co-localization with these markers and the Q dots, which were our stem cells. We did not see much of this at all, which is good, and that proves to us that most likely these cells, between this data and the Ki67 data, it seems these cells are going in there and they're kind of just sitting there. And then on top of that, we wanted to stain for, we wanted to see what type of reactivity there might be in the brain. So we stained for GFAP, which is gliofibrillary acidic protein, which is a marker of reactive astrocytes. We wanted to stain for IBA1, which is a reactive microglia marker. So it's been shown that there's a much increased staining with all types of brain injuries, whether it be stroke, whether it be TBI, or Parkinson's. So we wanted to see, how was this changing based on our stroke, and then how is it changing based on administration of stem cells following the stroke? What we're showing here on the right is we can really see that it's pretty clear when we compare all of our groups and we compare the left side of the brain, which was the healthy side, the contralateral side, to our ipsilateral side, which is our stroke side of the brain. There was much increased reactivity from both microglia and astrocytes on the side that had received the stroke, regardless of injection of stem cells. So when we compare our MCA only control, these 2 bars on the left, they pretty much look the same as all the other groups. So that's interesting. This is a bit of data that sort of, like a lot of this field, it's the jury's still hung on it. Some people report similar findings to us. Some people report that there was increased staining, not in the controls and only in the experimental groups. And actually, if we parse this out, we look at the data in another way, where we actually compare the sides to where we actually just look at each side of the brain for each stain. We can really see that there is no significance there. There is no increase in reactivity after a stroke, or I'm sorry, after administration of these stem cells after the stroke. So it seems that the stroke itself is causing the increased reactivity. The injection of stem cells is not playing any role. And then we also wanted to look at how is synaptic plasticity changing. So, a paper was published about 1.5 years ago in the Journal of Neurosurgery showing that they did a similar study to us where they injected stem cells into rats, but they did it once, and then they also had a group of repetitive injections at 3 times, and they showed that there was a thickening of the corpus callosum, showing that there must be some sort of synaptic plasticity that's taking place, where cells from around the brain are coming over to this area of the stroke, or to the area of injection of stem cells in order to kind of aid in recovery. That's not something that we saw. So when we looked at, when we measured the length of the corpus callosum, or the width of the corpus callosum following administration of these stem cells, we saw no change really in all 3 of our experimental groups. So in conclusion, we found that these MSCs delivered to the stable stroke provide functional recovery and benefit. The AMSCs stick around for a long time, but they don't show any differentiation or tumorigenesis. There was increased glial reactivity following the stroke, regardless of if there were cells given or not. There was no change to the corpus callosum size, indicating perhaps less synaptic plasticity or no change to synaptic plasticity. But our biggest thing here was that we saw that these cells are safe and effective. None of our animals died. None of them were unable to tolerate it. We only saw that, they recovered. There was no change to appetite. There was no change to weight gain. They were safe and they were effective. So what's next? So what's really exciting is this study was a preclinical trial for what we're going to be starting at Jefferson in the coming, hopefully the coming year, together with our collaborators in Rome, Italy. We are currently writing up our investigational new drug submission to the FDA and to the IFA in Rome in order to begin delivering these cells to patients the exact same way that we did it in the animals. So patients can come in, patients that have never got TPA, never got a thrombectomy, they can come into the hospital, they can undergo surgery where they would be injected with these stem cells. And we're pretty confident that that this is going to provide a large benefit to these patients. It'll be the first of its kind to deliver these autologous mesenchymal stem cells into the brains of a chronic stroke patient. So like I said, there's one other group in Stanford that did this in California, and they saw a benefit, but theirs were genetically modified. And so we really believe that the added benefit of giving autologous or the patient's own cells is they don't have to go on immunosuppressants. They don't have to worry about perhaps getting sick after, because giving a transplant from a donor, bone marrow donor, you have the possibility of rejection. And so this would really take that away. So it's extremely exciting. So I want to talk a little bit about why this study was so exciting for us and how we think it changed a lot of the previous, thought process around these types of studies, looking at stroke and measuring infarct specifically. And so I have up here the method section of 2 previous papers showing how they calculate their size of infarct. And both did it the same way. You basically, you take this healthy side of the brain, you calculate the size of that. You then take the dead side of the brain, you calculate the size of that, and you subtract one from the other, and you've got your size of the infarct. The issue with that is you're doing this on slides. You're doing this on histological sections. When you put these sections on slides, they have the ability to elongate or stretch out, and you're not going to get an accurate reading. You're not going to get an accurate size. Not just that, but if you want to carry out a long-term study, you don't have the ability to look at the size of the infarct following the stroke, following injection, or at multiple time points. All you can do is look at the time point following harvest of the brain, following sacrifice of the animal. And so we purchased the MRI from Scintica about a year and a half ago. And what that basically gave us the ability to do, and this is the same rat I'm showing here. This gave us the ability to see an animal at all of our different time points and measure the stroke. So on the left here, you've got your animal, the hyper intensity, that bright spot on the right side of the animal or left side of the screen. The left side of the brain is the stroke. And then on the right here, you can see actually how we quantify that. And so what we're able to do is we're able to outline, using some software, we're able to outline the stroke. And we're able to do that for each slice of the brain. And then we can actually get a millimeter cubed volume of the stroke. And, at each time point, we're able to measure this, and we're able to see how the stroke might change. So when I showed before that we didn't see much change to the infarct size, except for that one group where we believe it was due to some low N. Other people, who aren't using MRI might not have this capability because they only have the ability to look at the end. And so you don't really see how things are changing throughout. And that's, really important. So this is our MRI here in our dedicated space over on this left image. On the right side of the left image, you can see this is our electronics cabinet, which connects to our MRI. It's nice and small. And we have the two bazookas. We have one that holds the rat and one that holds the mouse. And then, so here's another cool picture I wanted to show of the delivery, the day of delivery, bringing it in, super heavy machine. But it was really easy to bring in, everybody comes out, we've got a whole team coordinates the delivery. And we kind of just wheeled it up into our suite. We plugged it in, the guys who came to install set it up, and we were ready to go. And here's an image of me on day one. We were putting an animal into the machine and seeing how it works. So what's really nice about it is it's really, really, really easy to use. There's this button here at the bottom. You kind of just click, turns on, you power up the software, and it's good to go. You're able to change out the coil, or we call the bazooka here, to put into the machine really, really easily. You just unplug it, plug the other one in if you're going between rat or mouse. If we ever have any issues, it's really easy to reach, the people that we work with, like Kara or Tanya. They're super easy to get a hold of, and they're always super, happy to help. And what's really nice about this system in particular, and working with the people that made it, is they're open to critiques of the machine. And so, for us, we noticed that our coil, we were having trouble as the rat got larger and larger. We were having trouble getting the rat's head in. And so we told them this, and they're currently designing one that is bigger, that will able to house a rat, for a long-term study. If you wanted to, keep a rat around for a year and they get, they grow and grow and grow, you don't have to worry about that. What's also really cool about this is, when it was delivered, it was just highly intuitive. You open up, this is the first screen that pops open. You're able to see there's basically like a playlist on the left of all our different sequences that we've run. And this is just within the past week, is how much use this is getting. But basically we can come over here, we click add, pops this screen up, we can put in our ID of our animal, the orientation of the animal, which coil we're using. So if I click this, I can change from rat to mouse. And then our protocol set. So, at the beginning when this first came, we were able to set up the protocols that we wanted to use, whether it was a T1, a T2, flare, DWI, all different types of MR sequences that might be helpful in our research, for us specifically using the T1, the T2. And then once you're ready to go, this screen pops up. So super simple. You've got your scout images, which basically are a quick screenshot of what the animal looks like inside, just really quickly to see their orientation if you need to change anything. And those will pop up here in these three. And then, it just goes down the line of all your different sequences. So we do these three, the T2, the DWI, and the T1, and it just goes one after another. And you just hit, once you're ready to go, you hit start. You let it run. It's not the loudest machine. I mean, human MRIs are much, much louder. This one, makes some noise, but it's nothing overbearing or anything too crazy. And it's really easy to change your protocol parameters. So if you want to change the number of slices, the gap between the slices, the slice orientation, if you're looking at a sagittal or coronal, it really allows for easy changes to the protocols. And with that, I will conclude my talk. If anyone has any questions, I'd be happy to answer them.
Perfect. Thank you for the presentation, Max. We will now move on to the Q&A section. And we have the first question, which is, this research is very exciting. How long do you think it will be before this protocol can be applied to human patients?
Yeah, so that's the hard part, is getting them into humans is difficult, as it should be. We want this to be the safest thing possible to get into people. Our hope was to get this rolling, for our submission to the FDA by October of 22, so about 5 months ago. Unfortunately, that did not happen. Right now, we're currently writing up our submission to our FDA. And we're also working on a couple last minute tests. So specifically testing the cells themselves. So we're going through and doing practice runs in that bioreactor, getting human bone marrow from donors in the hospital, growing it up, and then sending it out for sterility testing. And so one of the biggest hurdles that we see that other teams might not have, using these genetically engineered cells, where they can grow a huge, large group of the cells, freeze them down, and you're ready to just put them all into the same patients, or all the same cells into different patients, is that each of ours is specifically tailored for each patient. And so we have to be able to harvest our cells from the bioreactor and take them to the operating room within 4 to 6 hours. That means we have to get sterility testing, we have to get different toxin testing, all done within those few hours. So right now, that's the biggest hurdle we're seeing. We're on a good road. We're finding, good partners to help us with this. There's really good industry companies out there that are there for this specific reason. So we're trying, to go as slow as possible in order to make sure that these cells are safe and effective. Once they come out of this bioreactor, we're getting runs that are consistently safe. They're consistently not contaminated. They're consistently the cells that we want. And so right now we're just sort of maximizing our protocol and trying to make sure that what we do when we get to the clinic is the safest possible.
Yeah, thank you for answering that question. We have another question, which is the following. Did you pre-scan so each animal was its own control?
Yeah, so let me pull back to one of my previous slides here. Here we go. So if we look at here, and so the same goes for the behavior, because we did behavior and MRI on the same day, but we did do MRIs on the first day. I'm actually not showing that here because on day -28, the day following the stroke, there's a significant amount of edema or swelling and fluid around the infarct itself. And so what we actually saw was the size of the infarct was significantly larger at that day after the stroke, as opposed to a month later when all that swelling goes down and infarct stabilizes. And so that's why right here, we're only showing going from day 0, because, looking at day -28, it just sort of, it's not really adding anything important. It's just, it would just be a much larger stroke due to all that swelling around. But yeah, we did run that on that day -28 to use as its own control.
Great, thank you. The next question is, where in the brain did you inject MSC after MCAO?
Yeah, great question. So we targeted the peri-infarct region. So peri meaning next to, infarct is this region of the stroke. And so on day 0, we took these MRIs the morning of injection. One of my team members who I worked with really closely, Kevin Hines, is a 6th-year neurosurgery resident at Jefferson here. Last year he was on his research year, and we worked on this project together. It was an added benefit that he's going into functional neurosurgery, which is along the lines of these implantables and injections. And so he's very well versed in picking out the correct spots and targeting those spots, picking out good trajectories. And so what him and I did together was we found the correct area right in between where the ventricle was here and the infarct. And so we really try to target these areas. And so we have a stereotactic apparatus for rats, similar to ones that'll be used in humans. That's basically a robotic arm that we can upload these images to, that we can then pick, this is where we want it to go, and that's where the needle places it. So to answer your question, the peri-infarct area, the region outlining the infarct. So we targeted the medial side of that too, the right here in between the infarct and the ventricles. And we did multiple trajectories from the anterior to the posterior as well.
Thanks again for answering the question. The next question is, very interesting talk. I have one question. If we use hematopoietic stem cells instead of MSCs, what will be the differences? I'm just curious.
Yeah, it's a great question. I'll be honest, I don't know as much about hematopoietic stem cells. I think there is some research possibly using those, but it'd be hard for me to answer right now, just because we're looking at the MSCs. There's been a lot more research basically looking at the MSCs specifically because they're the lineage that differentiates to neural tissue. And I think there's just a wider breadth of research being previously done, but it's not a bad idea to look at the HSCs as well.
Thanks, Max. The next question is, hi, Max. Thank you for sharing all this great information. Great work by you and your team. What has been the biggest challenge throughout the process?
Let's see. So the biggest challenge, I would say, in this animal study was really, timeline. We wanted to get this done as soon as possible. This project is actually being funded by a generous donor in Philadelphia who was treated at Jefferson for a stroke, and he wanted to see see some new way to treat the strokes that could be used. And this research started many years ago before I was brought on. And now that this donor is getting into his older age, we want to be able to get this going before he passes. And so I would say being able to do this quickly and efficiently was one of the difficulties. And I would say the biggest difficulty is translating it to the clinic, to the operating room in this scenario is, working with the FDA and making sure that all of our testing is done and that we've done everything the same way that we plan on doing in humans, and figuring out the correct dosing. That's probably been the most difficult part, because we really want to test and retest to make sure that when this does go into people, it is going to be the safest thing possible, so that we see, these cells don't have any contamination. The cells are the correct cell that we want. They're not differentiated. They're not further down their lineage. And so I would say, yeah, all the testing that we've had to do has probably been, the most difficult.
Thanks again, Max. The next question is is the CORX developed stem cells into nervous system cells? I'm not sure if I'm pronouncing that one correct.
Yeah, I'm not sure what that what corks is. I'm sorry. I'm not sure I understand the question.
Yeah. Okay, but then we'll move on to the next question, and maybe the person who asked the question can let us know if she meant he or she meant another word rather than CORX. So the next question is, why did you choose the MRI sequences that you chose?
Yeah, so we tried to mimic the same MRIs that would, that a patient would get when they come into the hospital after a stroke. So, usually a patient has a stroke. Let's say they come in quickly or they take a while to come in. You have to get imaging to assess the size of the lesion. And so there are a couple different sequences that you can do that are commonly used in the clinic. And so two of those are the T1s and the T2s. So the T1 is basically, it shows the inflammation or the lesion or anything fluid will be dark. Fatty substance, so brain is, it's myelinated, so it will come up a different color as to bone or as to liquid. And so in this case, with the T1, we see a very dark shade with inflammation or fluid, and so that you can see here. And then with the T2, it's basically the opposite of that. And so we see a very bright hyperintensity with the with fluid or with inflammation. And so it basically, you can see it gives a detailed picture. And so there are other MRI sequences, and actually right now it's a big area of study is looking at different sequences for different for different things. And so yeah, these are two of the most commonly used. There's also diffusion weighted imaging, which is a DWI. But we found that the most clear picture that we got was from the T1s and the T2s. And the T2 it specifically is, we can see, it gives us the best ability to quantify these lesions in order to make assessments on the size of it.
Great, thanks for the answer. The next question is, what about the transdifferentiation of these stem cells becoming downstream lineage cells as adipocytes, osteoblasts, and chondrocytes?
Yeah, so one of the biggest questions too is, these are stem cells. Stem cells break off and become other types of cells. When you're a developing fetus, you have three different cell types that come out of that blastocyst, and that will become different tissues in your body. And so with mesenchymal stem cells, you have they mostly become chondrocytes, which turn into you have adipocytes, which turn into fat, osteoblasts, which turn into bone, chondrocytes, which are like a connective tissue or cartilage. And so one of the most important things is to look at, are these cells staying stem cells when they put into the brain? So the first thing we did was we did flow cytometry. There's a handful of markers that basically you have these positive markers and you have a couple negative markers. And when they're all shown together, that we have a mesenchymal stem cell. It's been widely published and there's actually guidelines for it. Because we're growing everything up in a bioreactor, we have optimize the amount of time that it takes for them to grow into this specific type of cell that they are, so that they are not differentiating downstream. And so we were very confident when we put them into the brain. But one of the things we also noticed was that since we left them in for a while and they seem to stay for a while, we wanted to make sure that they're not differentiating when they're in the brain. And so right now we're actually doing those studies. We haven't totally finished that up yet, where we're actually staining our brains for these markers for those different types of cells, the adipocyte, fat cells, chondrocytes, or the cartilage cells, or the osteoblasts, or the bone cells, to make sure that these aren't going to be growing when they actually are injected. So we haven't totally finished it up, but I will say we have not found any. So it really seems that when we put these cells in, they're staying there, they're reducing the inflammation, they're lowering the pro-inflammatory cytokines, increasing the anti-inflammatory cytokines, and overall reactivity of the brain is staying level. And that's adding to the functional recovery. There's still a lot of work to done exactly what is happening. We have we have those answers that I just explained, but the jury's still out on what exactly is causing this benefit, as opposed to just the cytokine change or growth marker change. So yeah, to answer the question, in short, we are not seeing any downstream lineage transdifferentiation once implanted into the brain.
Okay, thank you, Max. We have time for one more question. And the question is the following. Which strain and sex of rats did you use for the MCAO?
Yeah, fantastic question. And this is a good one because we used male. And one of the big questions when we've been approaching the FDA is, well, how do we know this is going to work in females if you're just testing males? Testing female rats specifically is difficult. With their estrogen cycles, you get massive changes that would sort of skew our results. And it also, it just seems like the easiest way to currently do it is to test in males. That way you don't have to deal with all of the cycle issues with the female rats. The next step in this is to test female rats, and we're currently developing protocols in order to do that. But that is really one of the more difficult things to do. And so if you look at a lot of the preclinical research that's been done, it's mostly done in male rats. But we don't really believe that it will cause too much of an issue. I mean, the biggest issue for us was the bone marrow, is the change in bone marrow following the estradiol cycles in the female rats. And so when testing the male rats, you sort of get around that. But we believe that once we start to inject them into patients, this shouldn't really be a problem.
Great, thank you for responding to that question. I want to let everyone know that we've reached the end of our session for today, and to be respectful of everyone's time, we're going to wrap things up. As mentioned at the start of our webinar, we will be sure to answer any questions in a written transcript, and we'll work to get this out to you over the next few days. I would like to thank Max for the great presentation today, and I trust that we have been able to provide you with a better understanding of intracerebral transplantation of autologous bone marrow stem cells produces functional recovery in rats with long-term stable strokes. If in the coming days or weeks you have more questions about the modalities discussed today, I encourage you to contact us here at Scintica, and we'll be more than happy to assist you. Thanks again to all of you for taking the time out of your day for attending our session, and we look forward to seeing you at a future Scintica event. Have a great day, and thank you, Max, again, for the great presentation.
Thank you.